Bismillah ar-Rahman ar-Rahim. Hello and welcome to the third lecture of your online immunology course. Today we will be talking about diagnosis of immunodeficiency diseases. Before the end of this lecture, we will be expected to understand the basic mechanisms underlying tests for assessing immunodeficiency. You should understand the basic principle underlying flow cytometry, a very important modern day technique, and then to be able to determine the results of described lab tests. As you know, immunodeficiency diseases are diseases associated with a defect in one or more elements of the immune system. Therefore, to diagnose immunodeficiency diseases, this usually involves testing for deficiency in numbers or function of the key cells and mediators of the immune system, such as phagocytic cells, T lymphocytes, B lymphocytes, and complement. Therefore, for the purpose of simplicity, we basically divide immunological diagnosis of immunodeficiency diseases into four basic categories: tests for phagocytic cells, tests for complement, tests for B lymphocytes, and tests for T lymphocytes. For each one of them, we divide them into tests determining numbers or quantity, and then tests determining the function. Let's start off with tests for phagocytic cells. And as we said, the first item would be tests for the number of phagocytic cells. We can simply start by determining total and differential leukocytic count, which is basically the total number of white blood cells, including granulocytes and non-granulocytes. Granulocytes can be, for example, neutrophils, eosinophils, and basophils, or non-granulocytes, including monocytes and lymphocytes. Now, the number of monocytes, and you do remember that monocytes are the precursors of macrophages, which are very important phagocytic cells. The number of monocytes can be assayed by flow cytometry using fluorescein-labeled monoclonal antibodies directed against a specific marker that identifies monocytes, namely CD14. Now, if we go on to now the immunological diagnosis of the various immunodeficiency diseases, and I remind you that now we are testing for the functions of phagocytic cells. So what are the functions of phagocytic cells? If you remember from your basic immunology lectures, phagocytic cells will be recruited to a site where there is an antigen. They will adhere to the endothelium to enter the bloodstream to the infection site. And they will then reach the antigen and will phagocytose or engulf the antigen and destroy it by oxidative or non-oxidative killing. Now, taking that into perspective, the cell therefore uses molecular oxygen to produce reactive oxygen intermediates such as superoxide ions and hydrogen peroxide. All these substances are toxic to microorganisms, and this process is called, as you remember, oxidative burst. So let's first test for random motility and chemotaxis, the ability of these macrophages to go through the endothelial cells to the sites of infection. This is done by the modified Boyden chamber assay. And this is based on a chamber of two compartments separated by microporous membrane. The tested cells are placed in the upper compartment, and a chemotactic chemical substance is put in the lower compartment. After an appropriate incubation time, the membrane between the two compartments is fixed and stained, and the number of cells that have migrated to the lower compartment side of the membrane is determined. Now, how about testing for the ability of the macrophage to ingest? Testing for ingestion is done by incubating the white blood cells, which include the macrophages and neutrophils and other phagocytic cells, with heat-killed yeast or Staph aureus, and counting the ingested particles by a microscope, because these are very large and so they can be counted easily with a microscope. Finally, we can test for the function of intracellular killing of macrophages. Defects in oxygen-dependent intracellular killing, as in congenital, for example, NADPH oxidase deficiency, is detected by nitro-blue tetrazolium test. In this test, a blood sample is taken from the patient. Neutrophils are isolated from blood and stimulated with lipopolysaccharide, the receptor for which are toll-like receptors, to produce superoxide. Then the NBT dye is added. Cells with normal phagocytic function will produce superoxide with reduction of the colorless NBT to blue insoluble crystals that are readily detected within the phagocyte macroscopically. In chronic granulomatous disease, the phagocytes will not turn blue. Next, we go on to detecting certain defects in complement. Again, we either measure amounts, defects in the amount, or in the actual function. Measurement of the amounts of individual complement components can be done by any one of the many techniques that you took in Chapter 2, such as radial immunodiffusion or ELISA, to determine the amounts of complement. As for the functional assay of complement, the assay here is called LD50. The assay of the hemolytic activity of complement is performed by preparation of serial dilutions of the patient's serum, and then antibody-coated red blood cells are added to each dilution. After incubation, the serum dilution of the patient giving 50% hemolysis, detected by optical density, is determined and compared to a standard. We now move on to tests for B lymphocytes. Again, we go with the system of testing for first defects in number and then defects in function. How do we test for the number of B lymphocytes? In the peripheral blood and the lymph node biopsies, we do this by, again, flow cytometry. This time using fluorescein-labeled monoclonal antibodies directed against certain specific B cell surface markers. Two very important B cell surface markers are CD19 and CD20. So we use antibodies to CD19 and CD20 fluorescently labeled. How about the tests for the function of B lymphocytes? What is the main function of B lymphocytes? The main function of B lymphocytes is the production, of course, of antibodies. Serum levels of antibodies, just like any soluble proteins, are evaluated by ELISA, radial immunodiffusion, immunoelectrophoresis, any, again, of the various mechanisms that you took in Chapter 2. Finally, to detect tests for T lymphocyte function and number, we start by testing for the number of T lymphocytes. You can count mature T lymphocytes by flow cytometry again, using fluorescein-labeled monoclonal antibodies against T cell surface markers. What is the main T cell surface marker? CD3. So you use anti-CD3 antibodies that are fluorescently labeled in a flow cytometer to determine the amount of T cells in a certain population of lymphocytes. We can also count T cell subsets, CD4 positive and CD8 positive and so on, using fluorescein-labeled anti-CD4 and anti-CD8 antibodies. To test for the function of T lymphocytes, well, we have two major groups of T lymphocytes. We have CD8 positive cytotoxic T lymphocytes, and we have T helper or CD4 positive T lymphocytes. Remember, the function of T helper cells basically is that when the T cells recognize their antigens, they will proliferate, they will mediate a cell-mediated immune response, and they will release cytokines. So we test primarily by delayed hypersensitivity skin testing using candidin antigen or tuberculin antigen. These are intracellular antigens that necessitate the mounting of a cell-mediated immune response as opposed to a humoral immune response. The absence of reaction suggests impairment of cell-mediated immunity. We can also assess the levels of circulating cytokines, which are released by CD4 helper T cells, again by means of various mechanisms, including, for example, ELISA. We can also assess lymphocyte proliferation. We said this is an important function of T lymphocytes in an immune response. Antigen-specific T cell proliferation is used frequently as an assay of T cell responses. Basically, T cells from mice or humans that have been immunized with an antigen, antigen A, for example, will proliferate when they are exposed to antigen A and antigen-presenting cells, but not when cultured with unrelated antigens to which the host has not been previously immunized, such as, for example, antigen B. Proliferation can be measured by incorporation of tritiated thymidine into the DNA of the actively dividing cells. And antigen-specific proliferation is a hallmark of specific CD4 T cell immunity. To test for cytotoxic T lymphocytes, well, cytotoxic T cell activity is often assessed by chromium released from labeled target cells displaying foreign MHC class I antigens. Remember that the main function of cytotoxic T lymphocytes, CD8 positive T lymphocytes, is to attack and destroy target cells that are expressing the appropriate MHC class 1 molecule. The target cells are labeled with radioactive chromium. They are washed to remove any excess radioactivity, and they are exposed to the cytotoxic cells. The cell destruction is measured by the release of the radioactive chromium into the medium after the cell is destroyed, the target cell is destroyed, and this is detectable within 4 hours of mixing the target cells with the T cells. Thank you very much for your attention. You will now go on to answering the quiz for this chapter, and remember, this is summative, so it will be added to your overall continuous assessment grade. Thank you.